Let's begin with the oxidative phosphorylation. That is the fourth step of the aerobic respiration. Last time we are discussing Krebs cycles. That ends a product of Krebs cycle is 2 ATP molecules from 1 glucose molecules, although one Krebs cycle produced 1 ATP. And there is 6 reduced NADP and 2 reduced FAD are produced. So these are the three compounds: ATP, reduced NAD, and reduced FAD. They are being used in the next step, you can say. So ATP is the currency, so it is used to produce the energy, while the reduced NAD and reduced FAD they will diffuse to the inner membrane of the mitochondria. The inner membrane of the mitochondria is crystal. It is a specialized membrane which contains a protein complex that is also known as respiratory complex. As you can see in this diagram, we can highlight this as these are the some of the proteins here. So this one is the respiratory complex here, right? And it has a special name also, that is cytochrome complex. Cytochrome is one of the important protein of the mitochondria inner membrane. So that's why its name is cytochrome complex. So what is happening in the oxidative phosphorylation? The first step is the oxidation of reduced NAD and reduced FAD. This oxidation occurs at the inner membrane of the crystal. Here are some enzymes which are being fixed in the crystal, that is the inner membrane. These enzymes are NAD dehydrogenase and FAD dehydrogenase, which oxidize these two compounds to form protons, electrons, and NAD and FAD are set free. These protons and electrons are then moved in the proteins. Electrons move in the electron transport chain like this, from here to here. In this, the electrons move from high energy level to low energy level and release energy. And this energy will be utilized to pump the hydrogen ions from matrix to the intermembrane space here. This one is the intermembrane space in between the two membrane of the mitochondria. So these are two steps of oxidative phosphorylation. One is the oxidation of the reduced NAD and oxidation of the reduced FAD by the help of reduced NAD dehydrogenase and FAD dehydrogenase enzymes, during which protons and electrons are being produced. Electrons move in the ETC and release energy, and that energy is used to pump the hydrogen ions into the intermembrane space of the mitochondria. The third point of oxidative phosphorylation, is the hydrogen ions which are being pumped through the carrier proteins, that is hydrogen pump. This hydrogen ions concentration is increased in the inner membrane or intermembrane space. So this one is the intermembrane space, and we can highlight this here, intermembrane space, that is in between the outer membrane of the mitochondria and the inner membrane, that is crystal. So when these hydrogen ions are increased here, they are diffusing back into the matrix, diffusing back through a channel. This one is a channel protein, and they are diffusing back due to the concentration gradient. As there is a high concentration now in the intermembrane space and low concentration in the matrix, so that's why facilitated diffusions occur through this channel proteins. This channel proteins has ATP synthase here at this position. ATP synthase is an enzyme. This enzyme is attached on this channel protein here, and this helps in converting the ADP into ATP. and for every 3 hydrogen ions 1 ATP molecules will be produced, because this channel proteins is also a rotatory protein. It rotate. In a rotation 3 hydrogen ions can move. So actually these hydrogen ions cause this rotation, during which one time ADP attach here, then one phosphate attach here, and third is the ATP produced. So this is the click, click, click. So 3 hydrogen ions move to round this click to form ATP molecules, and this is oxidation and reduction reaction, and this one is known as chemiosmosis. So chemiosmosis is the production of ATP from ADP and phosphate by the movement of these hydrogen ions from intermembrane space to the matrix. Now the fourth point of this oxidative respiration photophosphorylation is these electrons also arrive over there. Now protons are also over there. Remember, these protons or electrons are being carried from the glycolysis, from the interlink reactions and the Krebs cycles. So now they have the position in the matrix now after producing ATP. So here comes the oxygen now. Remember, oxygen is not used in Krebs cycle, not in the link reaction even, and even there is no use of oxygen in the glycolysis. But in this fourth step of aerobic respiration during oxidative phosphorylation, this is known as OP, oxidative phosphorylation, and we are saying that here oxygen is being required. So before this, you have learned that during SLP in Krebs cycle, ATP is being produced when succinate is changing into succinyl. So here again ATP is produced, but this time oxygen is necessary for that. So this is known as oxidative phosphorylation. OP, I can abbreviate it, that oxidative phosphorylation. So how it help in production of rapid ATP production? Oxygen combined with these protons and electrons to form water. Water is being produced here, right? And this is one of the reasons that oxygen is also known as a final acceptor, because it is a permanent compound now, not like reduced NAD and reduced FAD, which is being oxidized over there. So water is a permanent compound which is being produced here, and it is a waste also. I mean, it will be removed from here. It is a respiratory waste and metabolic waste also. Therefore, we also known as excretion. Water is excretion because it is produced by a metabolic reaction. So hydrogen and electrons are permanently combined with oxygen. This will also help in reduction or reduce the proton concentration in the matrix. Therefore, more and more protons, hydrogen ions, move from intermembrane space to the matrix through this ATP synthase. So this enzyme, we are saying that it is ATP synthase here. ATP synthase. ATP synthase. So ATP synthase means the enzyme which help in formation of ADP when combined with phosphate to form ATP. And we are saying that it is a chemiosmosis process because it is due to the hydrogen ions movement in the rotatory protein or this channel protein, which is also known as ATP synthase now. And then we are saying that oxygen is the final acceptor of these protons, because it formed a compound, permanent compound water, which is also a waste of the respiration, aerobic respiration. And then we are also saying that hydrogen ions continue to move through this channel protein or ATP synthase to produce ATP energy again and again. A lot of ATP is being produced here. For every 3 protons, 1 ATP is being generated. Now, if oxygen is not here, it is a special case. If oxygen is not here, I mean when oxygen supply is cut off, during anaerobic respiration. So these protons, electrons no longer be combining with the oxygen because it is not here. So the concentration of the protons will be increased in the matrix, and there will be no concentration gradient set up. And the protons, I mean the hydrogen ions, will not move through this channel protein by facilitated diffusion, and ATP production stop. So if the ATP production stop, the concentration of the protons in the intermembrane space also increases. So no more oxidation of reduced NAD also. So this reduced NAD will no more oxidize, no more oxidation of reduced NAD and reduced FAD. So there is no free NAD and FAD is available. The role of NAD and FAD is to carry the protons from glycolysis, from link reaction, and from the Krebs cycle. Now if they are not available, no more protons will be coming from those Krebs cycles or link reaction or glycolysis. So in the mitochondria, Krebs cycles will eventually stop if oxygen is not available, because there is no free NAD and FAD available, and there is high concentration of protons in the matrix as well as in the intermembrane space. So no gradient is set up, so no movement of protons and no ATP will be produced. So eventually the Krebs cycles and link reaction will also stop, and glycolysis, you can say, may also stop, but it continues because in limited supply of oxygen or cut-off supply of oxygen, anaerobic respiration is continue. We will go for the detail of anaerobic respiration, why glycolysis continue in absence of oxygen, so later in the next lesson. So here comes now the summary of this oxidative phosphorylation, that how many ATP is being produced here. From one reduced NAD, 3 ATP can be produced, on average 2.5. One reduced FAD can produce 1.5 ATP, but as a whole we can say 2 ATP molecules are produced. So now we have to look at how many reduced NAD are being present over here. We can check this. If one reduced NAD can produce one reduced NAD can produce 3 ATP molecules, then we have total 10 reduced NAD up till now from glycolysis, from link reaction, and from Krebs cycles. So from these 10 reduced NAD, total 30 ATP molecules can be generated here. For example, from the Krebs cycle, 6 reduced NAD is there, so 18 ATP can be produced from the Krebs cycle reduced NAD, and 2 reduced NAD from the link reaction, so 6 more, so it becomes 18 plus 6, 24, and 2 reduced NAD, I mean 4 reduced NAD from glycolysis, then we can say we have more ATP molecules can be produced. So this picture, this table, we can look at glycolysis have net 2 ATP are being produced here, and from Krebs cycle 2 ATP is produced here, and from oxidative phosphorylation 28 ATP molecules are being produced. So as a whole, one glucose molecules can generate total net gain 32 ATP. So total ATP can be possible 34, but as 2 ATP are being used during glycolysis in the phosphorylation of the glucose to form fructose phosphate and fructose bisphosphate, so 2 ATP can be minus from 34, then net gain is 32 ATP. So it can be range from 32 to 30. here are some of the questions from oxidative phosphorylation. The product of oxidative phosphorylations we have discussed, there are 28 ATP molecules are being produced by OP in oxidative phosphorylation from reduced NAD and reduced FAD. And at the end, the NAD and FAD are set free. So there is a question that what is the role of this NAD and FAD? Their role is very much clear, that they are carrying the protons from different stages of the aerobic respiration. So here are some of the enlist. So number one, they are acting the coenzyme to dehydrogenase the removal of protons from different stages of the aerobic respiration, and they are carrying the protons and electrons as they combine with protons. So electrons also combine with it, and they form reduced NAD and reduced FAD. So they are meant to be reduced, right? So they are carrying electrons to the cristae and protons also, where they are becoming oxidized, and then protons and electrons are being released, and then NAD and FAD are set free. And these NAD and FAD, when set free, they are again available for carrying the protons and electrons from glycolysis, link reaction, and the Krebs cycles. The next question is here, what is the electron transport chain? So electron transport chain consists of mainly membrane proteins of the inner membrane, that is also known as cristae, that are the highly specific and sequential. Only the specific protein can transport to the specific ions. For example, the electrons are moving from high energy level to the low energy level proteins. One of the proteins is cytochrome. That's why it is also known as cytochrome complex. While the protons pump, that is carrier of protons, is being transported protons from the matrix to the inner membrane, intermembrane space, that is active transport. So these are the few transport proteins here in the cristae or cristae in the mitochondria. So next one is what is chemiosmosis. So chemiosmosis, as we told before, that when protons move from intermembrane space to the matrix through ATP synthase, that is the channel protein, then ATP synthase convert ADP and phosphate into ATP through oxidation reduction reaction, that is the redox reaction, and this is also known as chemiosmosis. So the ATP formation at the cristae is chemiosmosis. So what is the role of oxygen? So oxygen is acting as final acceptor of the protons and electrons, because they are being carried through their carriers NAD and FAD from Krebs cycles and different stages. So here they permanently combining with the oxygen to form waste product, that is water. So what happens when there is no oxygen? So electron transport chain will be stopped immediately, because there is no movement of protons from the intermembrane space to the matrix. The reason is that there is no gradient, maintained concentration gradient. So there is almost equal concentration of protons in the intermembrane space and the matrix, so no more movement of protons. So ATP is being generated as the 3 protons are being moved through the ATP synthase, so that's why the electrons also stop moving from that electron transport chain. So there is no chemiosmosis at all, so no ATP will be generated. So oxidative phosphorylation is the main process is being stopped here, and 28 ATP molecules will not be no more producing. So only glycolysis will continue due to some of the enzymes which are present in the cytoplasm. So what is substrate level phosphorylation, and what is the oxidative phosphorylation? If the oxidative phosphorylation stop due to the absence of oxygen, the substrate level will also stop, because the reduced NAD and reduced FAD will not be available, set free, so they are not available for the Krebs cycles to carry the protons. So SLP, that is being in the Krebs cycles and the glycolysis, will also be stopped. So reduced NAD stays reduced, so no free NAD and no free FAD is there. So dehydrogenase cannot work. Dehydrogenase enzymes are present in the inner membrane cristae. So no oxidation of reduced NAD and reduced FAD, no link reaction, and no Krebs cycle. So this is happening when oxygen is not available, right? So 3 to 4 marks questions will be in the exam paper, and 3 to 4 marks is the role of the oxygen, and chemiosmosis 2 marks or 3 marks questions can be asked. So reduced FAD 3 to 4 marks questions can be asked. So you have to describe them and explain them briefly. So this is all about oxidative phosphorylation during aerobic respiration. In the next lesson, we will continue with the anaerobic respiration conditions. So thank you.